If you're trying to figure out whether Fast Blue or BDA is the best tracer for your experiment, the answer is nuanced. They're often compared because both label neural connections, yet they provide researchers with different anatomical evidence. The decision ultimately depends on the readout your experiment needs. In some studies, the top priority is identifying the neurons projecting to a target region, and for other studies, it's following pathway structure through the tissue. Depending on the workflow, complementary tracing approaches may be needed. Fast Blue is commonly used when researchers need to identify which neurons connect to a defined target region. After uptake, the tracer is transported back toward labeled neuron cell bodies, where it can be visualized by fluorescence microscopy. That makes Fast Blue useful when the main goal is to identify labeled neurons or compare cell populations across samples. Biotinylated Dextran Amine, or BDA, is commonly used when the experiment needs a structural view of the pathway. Instead of focusing mainly on labeled cell bodies, BDA helps researchers follow axons through tissue and examine trajectory, branching, and terminal fields. The tissue processing is different too. Fast Blue can often be viewed directly with fluorescence microscopy. BDA workflows usually involve additional development or detection steps, which can support detailed visualization of axons, terminals, and pathway architecture. Those differences matter before the protocol begins. Fast Blue and BDA can both support neural tracing, but the right fit depends on whether the study needs projection identity, pathway structure, or a complementary view of both. Polysciences recently published an article on Fast Blue and BDA in neuroanatomical tracing workflows, including how the study endpoint, tissue processing, and imaging method can guide tracer selection. If you're interested in learning more about how each tracer fits into different workflows, check out the link in the description for the full article.